In this chapter, we will discuss about membrane proteins. So up to now, we mainly discussed in this course about soluble proteins, so proteins that are folded in water. Now we will discuss about membrane proteins. So membrane proteins are in fact associated to membranes, and I will discuss about that in a few minutes. So here I will first introduce what are membrane proteins. In the next videos, we will see how we can predict or which kind of methods can be used to predict the localization of the membrane segment, so the segments of the protein that are inserted in the membrane. And finally, we will also discuss about the prediction of the 3D structure of membrane proteins. So, membrane proteins are in fact very important because they are associated to a very large number of important functions in the cell. If you analyze a genome, in general, membrane proteins account for approximately 30% of the proteins that are expressed in a cell. But if you focus on the PDB database, so the database that contains all the 3D structures that has been experimentally resolved, you will see that you have a very low number of resolved 3D structure for membrane proteins. So here you have a huge gap between the number of membrane proteins that are expressed in the cell and the number of membrane proteins for which you have the 3D structure. Here you have the number of structures that were present in the PDB in 2016. If you have a look now in 2020, you have approximately 3,000 different structures. And keep in mind that in total in the PDB database, you have more than 100,000 different structures. So that means that in fact the number of structures for membrane proteins is underrepresented in the PDB database. This is associated to some experimental difficulties. Indeed, it's quite difficult to obtain a crystal for membrane proteins. It's not impossible, but it's more difficult than for soluble proteins. It's sometimes also very difficult to overexpress membrane proteins, and so to obtain enough quantities to perform the different experiments to obtain finally the 3D structure and so on. If you have a look at the types of membrane proteins, in fact you can distinguish three different categories here. The first one are integral membrane proteins. So here these are transmembrane proteins that are permanently attached from one side. You have also peripheral membrane proteins who are temporarily attached to the membrane, and you have also some polypeptide toxins. So here what is important to understand is that the medium that is seen by these membrane proteins is very different compared to soluble proteins. So here let's consider that you have here a membrane. So this is a membrane, and you probably know that this membrane is composed by lipids. So here you have different lipids that compose this membrane. so on. So here you have lipids, and you know that these lipids are apolar molecules. Keep in mind that water is a polar molecule. So that means here that the medium in which the membrane proteins are inserted is an apolar medium, which is completely different compared to water, which is a polar medium. So here, when you have a membrane protein, for instance a transmembrane protein, this protein is in fact inserted in the membrane, and you see here that you have some transmembrane segments here in the membrane. Here you have different transmembrane segments, and you have also segments that are in contact with the extracellular here and intracellular medium. And in the extracellular medium you have of course water. In the intracellular medium you have also water. So you see that the loops that connect the different transmembrane segments are in water, and the transmembrane segments are in contact to lipids. So that means also that in fact the amino acids, the types of amino acids that are at the surface of these transmembrane segments can be hydrophobic amino acids and are in general more hydrophobic amino acids because lipids is an hydrophobic medium. So here you see that you have a very important difference between soluble proteins and membrane proteins. In the case of soluble proteins, at the surface you have mainly amino acids that are polar amino acids, and these polar amino acids like to be in contact to a polar molecule, which is water, the solvent. In the case of transmembrane proteins, in the region of the transmembrane segments, you have mainly hydrophobic amino acids at the surface, and this is very different compared to soluble proteins. And the reason is that here at the surface, the amino acids are in contact with lipids, which are hydrophobic apolar molecules. So, okay. So here, that was a short introduction about these membrane proteins. If you have now a look at the structure of these membrane proteins, you will see that in fact you have mainly two categories. You have a first category that corresponds to membrane proteins for which the transmembrane segments are helices. So that's the first category here. All the transmembrane segments are helices. They are called the helix bundles category. A second category is a category where all the transmembrane segments are beta strands, and these are the beta barrels category. At present time, we did not identify a membrane protein where you have a mix of helices and beta strands in the transmembrane region. But this could evolve because, as we have seen, in fact, you have only a very low number of 3D structure of membrane proteins that are resolved. What we will discuss in the next videos and in the next section is which kind of method can you develop to predict the localization of the transmembrane segment. And in general, we will see that helicoidal transmembrane segments are easier to predict than beta strands. And we will see that in fact the transmembrane beta strands are in general shorter than the transmembrane helices, and that's one reason why the transmembrane beta strands are more difficult to predict than the helices. And in general, these transmembrane beta strands are also less hydrophobic than a helicoidal segment.